In this video, you will learn step 3 of the PCR process, cDNA dilution. To begin, take out twice the number of thin-walled 0.2 ml PCR tubes as you have samples. Label 2 tubes for each sample. One tube will be a 1:5 dilution, and the other will be a 1:160 dilution. Here, 1:160 is shown in red, and 1:5 is shown in green. The dilutions used are specific to this experiment. This is the general outline of how the dilutions will be made. For a different experiment, the actual values may be different, but this procedure will remain constant. Take the cDNA samples out of the freezer and allow them to thaw on ice. First, pipette 72 microliters of Moby water into all of the 1:5 dilution tubes. Now, pipette 93 microliters of Moby water into all of the 1:160 dilution tubes. Your cDNA sample should now be thawed. Vortex the tubes on high speed for a few seconds to ensure proper mixing. Then, briefly spin the sample down with the MyFuge Mini to make sure that there is no sample caught in the lid. You are now ready to start diluting the samples. We have already completed the Moby water portion of this, so we will begin by pipetting 18 microliters of cDNA into each of the 1:5 dilution tubes. As you are pipetting the samples, discard the cDNA tubes. There is only 2 microliters left, and this cannot be used for further experiments. This will also help you keep track of which samples you have already pipetted. When you have finished pipetting all of the samples, vortex the 1:5 dilutions with the cDNA in them, and then centrifuge to remove the samples from the lid. Now, using 10 microliter pipette tips, pipette 3 microliters of the 1:5 dilution into the 1:160 dilution tubes. This time, you cannot discard the initial tube, so to keep track of which samples you have already pipetted, simply move them around the tray. As before, vortex all samples on high speeds for a few seconds, and then put them in the MyFuge Mini to spin down to make sure that there is no sample remaining on the lid. This completes the cDNA dilution step. Keep the samples on ice until you are ready to pipette the plate. To learn how to pipette the plate, please continue to the next video.